As a life science leader, your job is to make high-stakes decisions, often with incomplete data, tight budgets, and mounting pressure from investors, regulators, and payers. Scientifica Sessions cuts through the noise. Each episode brings you candid conversations with executives, innovators, and healthcare disruptors who've built functions from zero, scaled science-backed strategies, and navigated launches in some of the most complex therapeutic areas. Whether you're preparing for a regulatory milestone, integrating real-world evidence, or aligning medical with commercial, we provide you the insights to lead smarter, faster, and with clarity. We talk science, we talk business, we talk risk. If you're building something ambitious and you want to hear how others at the top are navigating uncertainty, competition, and growth, this podcast is your competitive edge. And that's just what you'll find on Scientifica Sessions. So press play. Let's get to work.
Welcome to Scientifica Sessions. I'm Tabari Baker, CEO of Kronos Scientifica. We are excited to have with us today Tom Neyarapally. He's the CEO and co-founder of Archetype Therapeutics, an AI-native company pioneering the use of generative chemogenomics in patient data-driven drug R&D in cancer and in other diseases. Previously, Tom was CCO and founding team member at Semaphore, a patient-centered health intelligence and genetic testing company that went public in 2021. He was also a member of the founding team and EVP of corporate development at the causal AI drug discovery company, Atia. After graduate school, Tom served as a corporate and IP lawyer at Chadbourne Park, LLP, and Frommer Lawrence and Hogg, LLP. He started his career after college as an analyst focused on pharmaceuticals at the management consulting firm Arthur D. Little. He earned a BS in chemical engineering from the University of Connecticut and a JD and an MBA from Cornell University. Tom, welcome to Scientifica Sessions.
Thanks, Tabari. Glad to be here.
Well, Tom, it's exciting to have you here on Scientifica Sessions. You have an impeccable background. You've been both in the corporate world and you've been involved in a number of startups. You're now doing something really novel and new, so new, in fact, that you've made up a phrase, generative chemogenomics. What is that?
Yeah, generative chemogenomics, given that generative AI is a hot topic and we are, in fact, using those types of capabilities in our platform. Generative chemogenomics refers to this concept that we can go from the chemical structure of a potential drug to its effect on biology as a whole. And the way that we measure that effect is in the genomic space. So rather than trying to understand how a molecule might bind a target, which is traditionally how drug development occurs, the concept with generative chemogenomics is trying to understand how a molecule will affect biology of a disease more broadly, and we measure that through the expression of a set of genes. So that's, as you said, a term, Tabari, that we coined. I hadn't seen it out there before when we first started using that term, and it captures the essence of the novel part of what we do. And as we get into it, I'll describe how that connects to patient data and how we use those things together to discover drugs.
Yeah, that's awesome. That's a much more systems approach to drug targeting, it sounds like, where you're looking to not just identify a singular target in the classical sense of drug discovery, but you're looking to understand system-wide. You may not even know the target, sounds like, but you're looking to see how a drug is going to affect the system. Am I getting that right?
That's right, at least at the outset of our process. And that point that you just made is really important as it trips some people up when they hear about what we're doing. This concept that, hey, you're not looking for targets leaves some wondering, well, what are you doing then? Because this is what drugs hit targets. Of course, drugs hit targets, and the drugs that we discover hit targets as well. But one of the limitations of looking at just one target is that, particularly in cancer, but also in many other diseases, the biology is more complicated than just what one target's effect is on the system. In fact, we have glimmers of what's happening in the biology through many markers. And so why throw all that out? Why not try to address all of that in the modeling effort? So by trying to learn the relationship between a proposed molecule and its effect on all of biology or a broader swath of biology, we are less likely to miss something. We're more likely to capture subtle, multiple pathway type situations and other things that you can't capture if you're just focused on, hey, how does this molecule bind to a single target?
Yeah, that's awesome. I think we do not know a priori all the time how drugs are working. We know a singular target usually because that's the way classical drug discovery has happened. But I think about the example of metformin, for example, how that drug, initially designed for diabetes, has now been maybe used as a quasi wellness drug because it's shown to have efficacy in other disease states. Is that what you're trying to get at with your kind of screens and your generative chemogenomics?
Yeah, it's one of the things. So talk about metformin or talk about one of the hottest topics in our industry, GLP-1 inhibitors. And the way we think about it is, of course, the GLP-1 targeting doesn't magically solve everything. There are things that happen downstream of that that then play out in all these different tissues. And so one possibility is we'll just skip to that answer, look downstream of that hot target, understand what's happening biologically, and find ways to target those things, perhaps with different drugs or perhaps with an oral small molecule instead of an injectable and these other types of things. So that is one part, although not the totality of what we're addressing. I think it's one really important part of it is that we're able to get to the downstream action rather than just focusing on the upstream purported target. I use that term a lot because, as you said, for development purposes and for perhaps investment purposes, people need to have the confidence that they know what the target is. But in reality, they may not know the actual target, or they may only know one of 5 targets that small molecule is hitting. Those are the types of things that we don't get tripped up on as much because we're not so focused on that singular target.
Thank you for helping me to understand kind of the biological rationale for this method of identifying drugs and identifying drug targets. I'd love to pick your brain on how you believe that integrating clinical genomic data will enhance the predictive power of these virtual screens that you're doing.
Yeah, when you look at what all the AI drug companies are doing, and there's a new AI drug company perhaps every day now at this point. One of the questions I have, and when people ask us how we're different than these other companies, one of the questions is, are you starting with the patients who you're actually trying to solve a problem for, or are you starting somewhere else? And I'd argue if you're not starting with a defined cohort of patients with unmet medical need, you're likely to miss something. It goes back to the target discussion that we were having earlier. But by starting with, say, a TPP, a target product profile for that set of patients that you want to solve a problem for, then the things that you discover will be relevant to that particular patient population. And we can go downstream from there. So that connection by starting with the patient population, what we do is we say, we want a patient population that has that unmet medical need. We want to understand what their outcomes look like. We want to follow them from a clinical perspective. And we want genomic data from the tissue, the affected tissue, where the disease is playing out. That's what we mean by clinico-genomic data. There's clinical data and there's genomic data from sample. And that sort of thing's been happening for a long time. So think about Cologuard or Decipher or Oncotype DX. These are tests that take a sample from a patient, understand the genomic profile of that sample, and connect that to a clinical outcome. Time to death, time to recurrence. Are you going to have invasive cancer? Are you going to have non-invasive cancer? The outcomes are myriad in their complexity and differentiation. But the bottom line is we're looking for some connection between an outcome that matters that we're trying to solve for and the genomic data. So we can build classifiers like those commercial classifiers that I just mentioned. Those typically are used to understand what's going to happen to a patient. If you have this genomic profile from your disease tissue, you're going to have a good outcome. You have this other genomic profile, you're going to have a bad outcome. We use those in combination with that generative chemogenomics that we talked about. And what that allows us to do is to say, propose a molecule or a billion molecules. It could be, and that's the scale at which we're able to operate in a day, and go from that starting point, understand what that molecule will do to the genomics of the biology of the disease, and then answer using that patient data component, what would that mean for outcomes in a particular patient cohort? So chemical structure, effect on biology, and then effect on biology to clinical outcomes. When you put that together, you have chemical structure to outcomes. So you could take a billion molecules and get a prediction as to what that molecule would do, each one of those molecules would do to clinical outcomes in those patients in a day. And that's what we've done, and we'll get into that some more, but that's the concept kind of end to end from the clinicogenomic aspect and the generative chemogenomic aspect. That's how we put that together to do this massive virtual phenotypic screening.
That's awesome. I came up in the lab days where you did RNAi screens by hand,
Yep.
where you were pipetting into 384 well plates and looking at cell death. So being able to do this, one, with AI is phenomenal. Two, what's also different is that when I was doing this back in the day, we didn't have outcomes on any of the disease, right? So we were guessing as to what an outcome would be. The great thing that you guys have is you have that outcome already, so that or the proposed outcome that you want already in hand, and you know that a priori. So being able to screen a billion molecules faster and get to the designated or desired outcome is phenomenal. I love that. The question I have, though, is you're screening a billion molecules, let's say. You find 10, I'm just going to make it easy math for me, that have a signal. What's your next step? Or do you immediately go to a regulator and apply for an FDA clearance, or how does that work?
Perhaps in a bright, shiny future, we'll get there. We're not there today. What happens next is we have to put these molecules through the same paces that any small molecule in development needs to go through in preclinical development. That would start with in vitro assays and go to in vivo assays. Even that is evolving. We've heard from the FDA about the acceptance of perhaps organoids instead of animal models. And if those organoids are derived from human tissue, perhaps they're even more relevant than what an animal model, a genetically engineered mouse, might have to say. So watch that space. That's going to continue to evolve over time. But we will have our molecules subject to whatever the latest and greatest validation assays are that people use for traditional processes. The difference is they might take through 2 or 3 years to get to those 10 that you were talking about, maybe longer. That might be someone's career to get to a target that is anointed, and then people go after it. And then years later, there's 10 hits that they want to try to bring forward. This process for us, as we'll get into, weeks to months. So it's a very different process to get to those 10 that you were talking about, Tabari. And then you have to go through and validate them. We're not at the stage yet today where we'd say, well, we're so confident we're going to go into a patient next. But working closely with, and there's a lot of other innovations happening in that space. How do you validate these things quickly? How do you run a lab process quickly? How do you create a lab in the loop, connecting the type of stuff we're doing in the AI side with what happens in the lab? And we're talking to very interesting partners about doing that. So there's even more acceleration to be had. We accelerate the process of getting to that 10, but then even that process of validating the 10 and iterating on those 10 and improving those 10, those are all things that are also possible in partnership with other companies. It's not our goal to, within our little shop, solve all those problems, but we are very much out on the lookout. And for those that are listening, interested in partnering up with people that bring other parts of the puzzle that we're going to elucidate as we go through the discussion today.
Yeah, that's fantastic. So the rules are still the same. You still have to go through your in vivo and in vitro testing. You still have to submit through the FDA the as the rules say today. There's no shortcut on that front, but the shortcut is really, to your point, is a billion to 10, right? And 10 to testing as quickly as possible and validation as quickly as possible.
Yeah, and I would add one more thing, which is that it's a shortcut to get to 10, and most people can't handle a billion. So most people aren't starting with a billion. They're starting with 1,000, or they're starting with 100,000 in a high throughput screen. A billion is a number that, if you do the math, if you were to try to take a billion molecules, put them in a petri dish or put them in a 386 well plate like you were talking about, how many 386 well plates would you need to do that? Well, it turns out that one of our potential investors asked us to do the math and put it on a slide. You get into numbers that are so silly that I hesitate to even mention them on this podcast. Trillions of dollars to try to put those all in, generate gene expression, evaluate whether those are good gene expression patterns. You just can't do it. So we're not just creating shortcuts, we're opening up avenues that were just not possible at all before this type of approach came online.
That's great. I'd love to hear how innovation saves money, because I think that's always what a challenge is in our industry. Is we all have heard the quote, it takes $1 billion for an innovation to get from from an idea to the clinic, right? And into patients. If you can take 1 billion molecules and save trillions of dollars and get to that a lot faster, maybe it is still $1 billion spend, but that $1 billion spend is actually on the back side of it instead of on the front side, just getting to from the idea.
Absolutely. Yeah, that idea of that $1 billion or $2 billion number depends on who you ask. Where does that come from? It comes from the failures.
Yeah.
It doesn't actually cost $1 billion for each one, but what happens is they cast a really wide net in the beginning, but their wide net is a fraction of our net. So they might start from 10,000 or 100,000, and they winnow their way down. And as you said earlier, they're not starting in the context of patient data, so they're missing that whole context. They're just going after a target. And then they get all the way through the end of that preclinical process and get into the clinic. And in oncology, maybe there's a 90% failure rate. So that $1 billion or $2 billion is encompassing all those failures. And as you said, that money can get redistributed. If you're not taking all 10 of those shots because you know 9 of them are going to be failures, or they never even make it that far because you've already de-risked that failure, then, as you said, maybe it's 5, maybe it's 10 drugs that get developed instead of 1. The other way to look at that math is, instead of $2 billion, perhaps it's $200 million to get that drug at least to the clinic or through the clinic. So that's the opportunity with, as you said, with AI. It's not just about speed. Speed means money for pharma companies. As you know, if a company gets to market 3 months faster, that is hundreds of millions of dollars or more. If you're battling with someone else to be to market first, being first versus second could cost you years of market dominance. That's the market side of it. The patient side of it, and really that's the inspiration for what Paul and I are doing here at Archetype, is this idea that there are people that are just not getting medicines they need because various of these things that we're talking about are not being done, and they can be done. We now have the tools to do it, so let's go do it. That's what we're doing here.
Love that. Love that. I mean, it's a great mission. I think you guys are well positioned to be kind of that nucleating company for others to go off and kind of help to build or develop new innovations as well. So screening was like my friend in graduate school said, he's like, screening is where it is. And I was like, screening is okay. He's like, nope, this is where you actually find the things. So he is, and shout out to Casey, if you're listening, he would love to hear this conversation and be involved with this. So we've talked about kind of what you're doing at Archetype. We've talked about how the process works. We basically alluded to how your drug discovery and early stage discovery is sharper, right? Is more targeted because you can cast a wider net and you have a lot more information to narrow down the appropriate targets and or drugs, not necessarily targets. Love to hear an example of that. I think your lung cancer program is one that you have talked about recently. Could you share a little bit about that?
Absolutely, Tabari. Happy to. We were inspired to start work in early stage lung adenocarcinoma for a few reasons. One, lung cancer is the biggest cancer in the world. So there's a lot of unmet need in general. When you think about early stage lung cancer, if you can head that off before that becomes invasive and ultimately metastatic, you're going to have a lot more impact than if you're extending life by a couple months on a stage 4 patient. Those 2 months are super valuable. I wouldn't denigrate that. There's value to that, and it's valuable to society. It's valuable to those patients and their families. But it'd be a lot more valuable if we could save their lives by intervening early. That's not our idea. Experts at Mount Sinai in New York and our colleague and collaborator, Dr. Charles A. Powell, who goes by his initials, CAP, is an expert in this, has been working on this for a very long time as an academic. He also has a position in management at Mount Sinai, but his research interest is in this area. So that's beautiful for an AI company like us. Someone else has found a really meaty problem. They've profiled patients and collected some of that data that I talked about, that genomic data, that clinical data, and they've already done their own work on it. They've taken a run at trying to discover drugs using that type of data. One of the types of approaches that Paul, my co-founder, and I have worked heavily in, but that we're not using here, is called network or systems biology. That's the concept of trying to learn how all the pieces of the biological puzzle fit together using math, computing. The idea that gene A is upstream of gene B and downstream of that is gene C. And if I intervene at one of those points, I'm going to affect what happens downstream. That's what that type of modeling does. Often what it's done is in service of trying to discover a target. And then you fall into some of the limitations that I talked about before. So it's great. We're co-inventors on these types of technologies. We've done this stuff. We believe in the power of it. But we believe that you can leave some stuff on the cutting room floor if you go down that path. And so our collaborator, Cap, had gone down that path, and they discovered interesting, what they call hub nodes or key parts of the gene expression network, where the hypothesis would be, since that's so important, if I intervene there, I'm going to interrupt this disease from playing out. In this case, I'm going to stop cancer from becoming invasive and ultimately metastatic. So that's what they did. And they published in Nature Communications on that process of intervening with molecules that they identified because those molecules had targets that they thought were important. So they went out and found an Amgen drug that hit aurora kinase A and B, and they identified those two targets as important targets based on this network biology type work. And again, published in Nature Communications in 2022. And so when I reconnected with Cap about a year after that, I showed him what we're doing, what we've been talking about here on the podcast. And he said, that's very intriguing. I'm not sure how much I believe that could work, but I know you guys and I know you're smart guys. Let's give it a shot. Why don't you build your lung cancer in silico module, generate that 10 that you were talking about, take the billion to 10, throw the 10 over the fence to us. And we've got all the assays set up here at Mount Sinai to do this. We can very quickly tell you whether you're on the right track or not. He was intellectually curious. He was motivated by some of the things you were talking about, Tabari, that we can get some more drugs to patients faster and more efficiently. And so we set up this collaboration a little over a year ago, really not that long ago. Within a month and a half, we had built the computer models. So it didn't take us 3 years. It took us just weeks to do that. And we had the simulation. So once we built the models, we took large numbers of small molecules. virtual molecules that people have never even made before. So listeners might not be aware of this. There are virtual catalogs of billions of molecules. They're not sitting in vials on a shelf. They're sitting in a catalog offered by Enamine or E-Molecules or one of these types of companies, where you as a pharma company, whether you have 2 people or 20,000 people, it's democratizing. Anybody in any company can go to that catalog and say, I want number 2,300,491, and they'll ship it to you in 3 weeks for a few $100. So anybody can get access to that. Then it becomes a question of, well, who can actually digest that? Who can take the billions and do something with that? Well, the average pharma company and the average person in drug discovery can't do anything with that because they don't have any rational framework to sift through that and identify what could actually be useful. So that's what attracted CAP to working with us. We threw over to CAP, ultimately started with molecules, a smaller set. We started with molecules that had been to at least phase 2 clinical trials. And that's tens of thousands. So even that is a large number. And we identified molecules that we thought would be interesting based on what the in silico readout was. These molecules had a good in silico readout. They generated gene expression patterns in the computer that at different concentrations led to coherent dose response in terms of the clinical outcome. That's what our platform allows us to do. So we took the top 5 that we could quickly get our hands on, got them shipped to us, sent them over to Sinai, and said, put these in your in vitro assay first, then we'll go to the mice. And all 5 of them had an effect. Some were better than others, but all 5 of them, the chance that all 5 at random would have an effect in this specific cell type is limited.
Right.
Now, in vitro systems have major limitations, and we could talk about that more, but that was the starting point. We identified a MEK inhibitor that looked pretty interesting called mirdametinib. That MEK inhibitor is owned by SpringWorks Therapeutics, which is in the process of being purchased, and actually I think now has been purchased by EMD, Merck Serono, the other Merck, the non-US Merck. And it turns out that company also had another MEK1 and 2 inhibitor as well that they had out-licensed to Day One Pharmaceuticals, but they own now 2 MEK1 and 2 inhibitors. And our discovery really was that MEK1 and 2 inhibition might be really interesting, not in late-stage cancer, because that drug initially had been brought to development by Pfizer, the original owner, in late-stage lung cancer, and it failed. So one might think, well, it's not a good lung cancer drug. Our hypothesis is, no, it might be a great lung cancer drug, but only if you apply it at this point in the development of the disease. Early stage disease looks very different than late stage disease in terms of the pathways that are active, the options that are open to us to intervene in an effective way to stop that cancer from becoming invasive and spreading. So that finding was interesting, and it was interesting that 2 MEK1 and 2 inhibitors popped up at the top of our list. So going to your earlier question when you were saying, it's not targets, it's molecules, it's actually both. It's just that the way we get to the targets is not direct. We don't start with targets, but we can end up with interesting insights about targets. So our interesting insight is almost an indication expansion. I know later we're going to get into how does this tie to med affairs and other parts of these companies. Is this just a drug discovery? Not at all. The question of unmet need and the question of indication expansion, which is really a clinical development or even kind of commercial med affairs type question, is absolutely what we address here in part. And this is a real example. So we identified for someone else. It's not our drug. So really, because this was all presented publicly, obviously there's IP that we filed and so on. But bottom line, now it's out there that, hey, MEK1 and 2 inhibition might be interesting for intercepting early stage lung cancer. This was presented by Mount Sinai. I didn't even get to the in vivo part. Maybe perhaps I'll touch on that first. So in vitro, check, looked really interesting. So then we went into these genetically engineered mice that were genetically engineered specifically to model the development of invasive lung cancer. What's nice about these is these are not immunocompromised mice. So they have an intact immune system. That's one of the issues with, say, xenografts, for example, is you make a tumor grow on the back of a mouse. How much is that like real cancer development? Some would argue it's not really at all, but you can kind of shrink the tumor. And that's often what people want to see in late stage cancer, which is where most cancer development starts. So we did xenograft work that looked good as well, but I'll focus on the GEM model work. Genetically engineered mice, that this was an insertion of a KRAS G12D mutation.
Yep.
Many listeners who are in our field in cancer will recognize that mutation because there are drugs developed for that mutation, combined with a TGF-beta receptor 2 knockout. So those two modifications in these mice caused them to develop invasive lung cancer. Cap and his team had developed this model over many years at Mount Sinai, and I believe even in Columbia before that. So they used that model to test that Amgen drug that I talked about a few minutes ago. And they showed modest but appreciable and quantifiable reduction in invasive and lipidic tumors in these mice. Based on that, they were able to publish in Nature Communications, based on that difference. We're not looking at a picture of this here on this audio podcast, but we have a slide that was presented at AACR by Mount Sinai that showed a comparison of that result to control mice, which are just riddled with tumors. So their result was better. And then our result, which shows an almost pristine slice of a mouse's lung treated with mirdametinib. So our partners at Mount Sinai were blown away. And that's what was the impetus for that getting presented at AACR. They said, we were skeptical at first. We got more interested when we saw the in vitro data. And now seeing this in vivo data, they were excited about, hey, can an IIT be done based on this? What can we do with this? So that whole process that I described from we should go after this to the end was a handful of months, under 8 months to do all of that. And these are the types of results that if you were doing this in a department in a pharma company, that might have been 2 years and I don't know how many $1 million, $5 million, $10 million. So very rapidly getting that type of result is what we were able to do. And then we went further. We said, if that looks interesting, that drug mirdametinib looks interesting, what about other molecules that in silico look similar to mirdametinib? They create similar changes in biology. So we found some of those. We went back to Mount Sinai. You could probably guess what happened. Those worked as well. That was part of the AACR presentation. We then upped the ante further and said, let's take one of those library molecules and engineer in some more activity by predicting how that change in the structure of that molecule would influence gene expression and therefore clinical outcomes. And we were able to identify, we took one of those library molecules, we identified through millions of iterations of possible changes, 24 new molecules that look better in silico at changing the outcomes in the patients. We had the 24, we sent them to India to our synthesis partner and said, we know we're a small company, we have 2 slots with you guys. You pick 2 that you can make the quickest out of the 24. We're rushing to get this data in for AACR for the presentation in April. This was done over the holidays at the end of last year. The Indian firm picked the 2, shipped them back to us. It was a drum roll moment. Do these totally novel small molecules that we made up using the computer, do they work the way we think in gene expression space and ultimately in impacting the outcomes? And they did. We were able to engineer increased activity, which if you think about it, the implications are profound. This is a different way to do drug discovery and optimization. So when we think about anybody in a pharma company that's listening to this, this is not just massive virtual phenotypic screening that someone in early drug discovery does. We've touched on the application for indication expansion with a molecule you may already have. But this also touches on late preclinical, which is, hey, I have my hit, but I want to improve it. What is my context for improvement? Where our lens for that is, does it improve impact on gene expression, which in turn then would impact improvement in patients, so that when we go to the clinic with that improved molecule, it's not, oh, what's the patient population? It's no, we started with the patient population. We have a biomarker set for it. So we know what to look for early in the clinic. And this molecule has been optimized to optimize that biomarker set that goes with that patient population. So that's what was presented at AACR. And that validated not only the platform, but then we had these early lung assets that were developed in just months that look really good in these assays that our partners have been spending years deploying to try to find molecules to help these patients. So that's why I'm excited about it. Is it was fast, it was relevant, we were able to do it at scale, and we were able to do it for a vanishingly small cost. I don't even want to advertise it to give potential partners the wrong idea about it, but this did not cost tens of millions of dollars to do.
Yeah, that is phenomenal. I think if you just talk about the speed, right? 8 months is a blink of an eye in drug development. That is no time. You're still getting your team sorted out on what we're going to do over 8 months.
That's right.
And you've gone from an idea to an AACR oral presentation, meaning the data has been generated both in vitro and in vivo. You've done a number of tests with known molecules and generated new molecules to further
validate, I think, is at least part of it. Yeah.
Yeah, to further validate this new class, because it's really a new class of drugs. MEK1/2 inhibitors have not been on the market for various reasons. One, to your point earlier, maybe they were used in the wrong context, in late stage lung cancer instead of early. But then two, a lot of these molecules end up on shelves and never to be seen again. And what you're effectively doing is going to the library and making us go back and read the classics, right? Shakespeare is good. We have to read that again. And there may be a different interpretation of how that molecule can be used. So that's fascinating work. I'm super excited for you guys at Archetype Therapeutics. I'm sure that was a true drum roll scary moment when those molecules arrived in house.
Yep.
And you're looking at these 2 vials saying, all right, guys, this is the moment here.
Absolutely.
You've given the advantages of that. What were some of the risks that you were thinking about as the drum was rolling, right? And you're developing these molecules.
Yeah, I mean, and even if we rewind the tape a little bit on that, one thing I'll say, just to revisit the last point on MEK inhibition, MEK's a known target. Pfizer gave up on it, and then Pfizer licensed it to SpringWorks, which was created to pursue rare indications. And actually, there was an approval of mirdametinib, the molecule we were talking about, in NF1, in a rare disease. So in that particular case, I think the cases you were talking about absolutely are also opportunities where the drug is just completely shelved. The middle ground is it's not shelved, but they just gave up on that big lung cancer indication. They went after a really narrow indication. They changed the dose, has on-target toxicity, so they changed the dose. That solved that problem, and they went after a narrow indication. And we were able to show essentially that actually if you had just turned 10 degrees rather than 180 degrees, you could have been in early stage lung cancer with a massive unmet medical need being addressed. So that's just to clarify on that point. Speaking about the risks with what we're doing, the first objection that people raise is what we touched about at the beginning, which is, wait, what's the target? So I don't understand how this works if there's no target. And just to reemphasize and clarify, of course, there are targets. It's not magic. These are physical entities, these chemistries that do bind things in your body. That happens. Now, the question is, what do they bind? When do they bind them? How many do they bind? This is where people oversimplify or sometimes often flat out wrong about what they think is happening. So that's one thing to keep in mind. Risk number one, or at least perceived risk, is, hey, there's no targets in this whole approach. That's just not true. We just discovered that later in the process. So put that one to the side. The next question is, can you connect what you're modeling essentially in a model system? It's done in a computer, but essentially what we're modeling is, what does the chemistry do to the biology of a lung cancer cell? Mm-hmm. Well, a lung cancer cell is not all of cancer biology. When you think about cancer in particular, and again, our platform has been employed outside of cancer. I should clarify that for listeners. This is not a cancer-only platform or cancer-only discussion. We've had a lot of inbound interest from immunology and other areas recently, in fact. But the question ends up being, how do you translate from a prediction as to what a molecule does in a cell type to what happens in the whole disease? Because with cancer as one example, you have the tumor microenvironment. You have fibroblasts. You have CD8 positive exhausted T cells. You have all this other stuff you have other cells that are playing a role or not playing a role. So how do you
got a heterogeneous tumor itself?
You have a heterogeneous tumor. There are different clones. All this other stuff's happening. It's very complicated. But part of how we address that is we take that patient data to use the context. We basically, I didn't clarify this before, but the way we talk about it and why what might be perceived as a risk is, I would argue, a strength is half of our platform is learning the relationship between chemistries and what they do to biology. That's that generative chemogenomics stuff. The other half is, well, what is the biology we're actually targeting? And that comes from the patient data. That's where the ground truth is. It's in real patient data and real samples. And we will get better and better at characterizing those samples using multi-omic modalities. Spatial omics is hot now. There's all these other modalities that will get you more and more fine-grained about what's happening. So what might be perceived as a risk, which is this translation between preclinical models and human data, I would argue is a strength. The drumroll moment was, well, does it actually work? Was my argument right, or were those naysayers right? And we talked about the result that came out of that. So that idea of, does it work? The other thing early on that was a risk was, we proved this out first in prostate cancer. We were able to make predictions about clinical trials before the results read out. People were super impressed. We had this example of talazoparib, this PARP inhibitor that Pfizer had a success on, where we made a prediction that that drug would actually be successful relative to its peers in the PARP class in a particular population, a population that had both DDR positive patients and non-DDR positive patients. So that was a result that made a big splash at ASCO a couple of years ago. We predicted it in advance. So one of the risks was, hey, is this just a one-hit wonder where, yeah, your platform works, but it magically aligns to the problem of prostate cancer and has no applicability outside of prostate cancer? Well, we de-risk that by the lung cancer results that we talked about. So those are some of the risks. It's the translational piece. Do these pieces that you've put together actually fit together, or have you created a Frankenstein? That's one part. The second part is, does that Frankenstein, or we would say this beautiful thing that we've created, is that replicable in other diseases? And we've shown that. So yes, there are risks. Ultimately, all of this is only as good as the training data. I'd say that's the fundamental, foundational risk for any AI thing. For our listeners here, no matter, it doesn't have to be in drug discovery, it could be in anything. What is the risk? The risk is that the wrong data gives you wrong answers or creates bias if we don't have the right training data, the right starting population. Those are all things that are risks with respect to the effective use of this type of technology.
I am reminded of the phrase garbage in, garbage out, right?
Perfectly
Anyone who does
applicable.
any data analysis at a large scale, I did some of that sequencing work back in the day, and we would always say, if you give me bad DNA, right, you're not going to get the answer you're looking for. And it sounds like the same is applicable here, and will continue to be applicable for a long time, I would imagine, in the AI drug discovery space.
Absolutely.
So with that risk, where do you find clean, and I'm putting air quotes up here, clean outcomes data? Because that's the hard part. I think that's the hard part. I think the easy part is the AI-generated things, but that's hard.
It's very important, as we talked about, the lens for biological interpretation of all that we do, in our belief with the AI, is that human data. Fortunately, compared to 10 years ago, there are a plethora of companies that have popped up that do this. Some of them are companies that are diagnostic companies by what they do, but then the exhaust of what they do is this great data that we're talking about. It's high quality because it's being generated for clinical purposes already. So think about these, Tempus is just one example that comes to mind off the top of my head, but there are Tempus, Veracyte, I could go on down the list. Those companies all have different levels of savviness about making that data available for purposes other than diagnostics. And some of them are more hip to the idea that, hey, this data could be super valuable for drug discovery. And we've had discussions with a number of these companies, and we have relationships and even contracts with some of those companies to get access to that data. We like the fact that it's clinical grade data, research grade data. Also interesting, but you're going to have some limitations. Has it been validated? Has it been taken out of sample? Has it been put through a CLIA and CAP filter or not? So the good news, Tabari, is these companies really exist today to a much greater extent than they did even 5 or 10 years ago. So I don't think that's as much of a risk as it was when I was working in this sort of thing 10, 15 years ago.
Yeah, I think that's the big risk that I was envisioning is you have the platform, which is phenomenal, that can be plugged and played in various different disease state contexts. having the appropriate data for that particular disease state with enough patients that gives you a signal is where I saw the big risk. And it looks like, at least in cancer, I know if you think Tempest, Caris Foundation, those types of places that are doing CAP-CLIA genomic sequencing, that data is pretty pristine. In these other disease states, I think there's probably some work to do. I think in immunology is a phenomenal new space where immunologists have done a lot of this work for a long time, kind of in the dark, and it's now being brought to light how much data they've generated. That's a really complicated space and could really use a tool like this. Maybe you want to transition here to leading in this space, right? So AI and drug discovery is relatively new. I mean, you've been in this space probably for the majority of your career here, but that's maybe been about 10, 12 years. I think now we've gotten to the compute power where we can really move quickly in this AI space. As an executive, how do you think about building out the capabilities that enable the use of AI in drug pipelines?
Yeah. When I think about, for example, what a pharma company would need, and ultimately they have the money, they have the ability to develop these things at scale. The number one thing I would say is, and we just talked about it, is the fluency in the data. That is the raw materials for this. So if we were mining for gold, you need the mine site basically to start doing your mining. And in our world today in pharma, that's data. So that's internal and external. So if you're trying to solve a problem for patients that are non-responders to drug X, and you are the developer of drug X, that data may not be out there with Tempest or Caris or Foundation or somebody else. It may be sitting in your shop, and you got to generate it, and you have to recognize that data that might have been developed or generated in a clinical trial may be useful for R&D purposes. One of the challenges I saw, this is an organizational challenge, and it's been overcome, I think, to some extent, depending on the pharma company. But when I was working closely with pharma companies 15 years ago, 20 years ago, we would see, the perfect data for this is your failed phase 3 of fill-in-the-blank IO drug, or it wasn't IO drugs, they're a drug for ulcerative colitis, for example. I'm thinking of one company, failed phase 3. And the research folks wanted to tackle that disease. And we said, well, the perfect data is sitting over in your failed clinical trial. And it took a year plus to wrangle that data out of development, all the turf wars, all this other stuff. There's also sensitivity. Anything that's a failure, nobody wants to look at it. And of course, that's where all the learning is. In life and in pharma drug discovery, the learnings is in the failures, not in the successes. So that's an organizational thing that people need to get smarter about, and I think have, but there's probably room for improvement. The other thing that I think is important for whether it's a pharma company or us to be smart about, and we're lucky here to have this, is to have multilingual scientists and leaders. So it's no longer good to have one guy that knows the biology, one guy that knows the chemistry, one guy that knows the clinical science, and so on. You need at least some people. Some men and women need to have both of those or multiple of those types of skills in one person. Those people become the connectors and the hub nodes, if you will, to pull all the pieces together. Now at Archetype, we have Paul McDonough, who is a biochemist, so that's already biology and chemistry. He was an instrumentationalist, so he was doing NMR spectroscopist by training in his PhD. He then built instruments. They took an inkjet printer at Rosetta Informatics 25 years ago and said, I'm talking about a printer like you would use in your office. And they said, 4 heads on here for the black and the 3 main colors, primary colors. there's 4 bases. We can just spray the bases using so that people don't know this because all this stuff is old hat now, but the Agilent SurePrint technology originally came from Rosetta, and Paul was one of the developers of that. So, and then later he ended up being involved in a group at Rady Children's Hospital with Dr. Kingsmore, where they saved a bunch of NICU babies' lives by doing genomic profiling, whole genome sequencing overnight, and combining that with clinical data and reasoning over that. Why am I ticking off Paul's resume? Because there's very few people that have that diverse background, and he has worked in big pharma companies, and I think he did very well there because people said, wow, there's nobody that has this very diverse background, patient-facing stuff, drug discovery, internal-facing stuff, instrumentation, data science, biology, chemistry. That's a unicorn. So I'm lucky we have a unicorn here. But even if you don't have a unicorn, a thoroughbred horse, if not a unicorn, would still be valuable versus kind of, the donkey sitting over there somewhere. So there's levels of skill that we can have here. And I think that multilingual capability is super important in this AI world, more so than it was in the traditional linear process in drug discovery that has persisted from decades ago to very recently.
I like to say, and I'm teaching my kids this, is knowing a little bit about everything is okay. It's okay to be a generalist now.
Yes.
And I have a PhD, so I can go really deep in one thing. But for those who are listening, who are maybe looking to be in pharma or DIA here in the next generation, I think you'll need a baseline level of knowledge of something, right, that you know deeply. But do not ever be afraid of jumping into something that you don't know but have to learn very quickly. Everyone that I've talked to across my entire career has done that. Tom, you're one of those. You've jumped from one thing to another. I've jumped from one place to another and learned a whole different disease state. I think that ability to contextualize what you see based on where you've been broadens if you've been more places. And so I applaud you for finding a unicorn thoroughbred horse, because I think he's both, actually. But I would advise everyone to do the same. So what I'm hearing is that one of the capabilities that needs to be developed by companies, if they're looking to kind of do new drug development in this AI space, is to have these hub node people, these people who are cross-trained either within their own system or across multiple disease states, so that they have the visibility or have had visibility to multiple things and can bring that expertise to you. Is there any other particular capability that an executive should be thinking about?
Well, beyond those things, there are soft skills. There are a couple things that I think are critical that relate to what we just talked about. Even if a given person doesn't have that Paul unicorn thoroughbred set of backgrounds and skills that we talked about, there's a EQ side of that as well, which is humility, not having all the answers and knowing that other people are part of the puzzle. And that humility applies at an individual level. It also applies at a company level. So for us, we don't think we're going to build it all, or that our one platform is the only AI platform that matters for drug discovery and we've got it all figured out. We've heavily relied on partnering with external parties to be able to get things done. And I think that pharma companies will need to do that as well. Know what you do really well and do that for sure and be really, really good at that. And feel free to outsource or at least partner on a bunch of these other things. The partnering helps you learn, but it also helps you bring in capabilities that you don't have internally and perhaps would not be so smart or cost or time efficient to try to build internally.
So if we think internally, right, and I think about Archetype Therapeutics, where you are currently. So you've got drugs in the pipeline, right? You're going through your in vitro and in vivo validation processes for some of those drugs. On the exit of that is a commercialized product. Between those two steps is a chasm, right? It's the valley of death is what we call it, or the chasm. How would you bring in the appropriate capabilities as you're transitioning from kind of that R&D focus into a potential commercialization focus? Not saying that this is the strategy or anything like that, but just help me understand how you would think about that transition.
Yeah, it's really that partnership model that we were talking about. When you look at all these AI drug companies, and now there's a 10 plus year track record of what they've done. So not even speaking specifically about us, but even a little bit more broadly about the companies in this category. When people have asked what's their strategy really has been a combination of building their own pipeline and partnering out these assets at different stages along the way. You learn when you partner something out, you learn from your partners. They bring all those skills that you may not have as an AI drug developer to the table. So that's really how one can do this. Now, as you said, I'm not speaking specifically to our business model, but more broadly about the companies in my sector. That's how I would think about it, is that the partnership allows you to extend yourself and what you've discovered and the value you've created beyond your own internal capabilities. The other way to try to do it would be to somehow magically hire all those people, all the right skill sets internally, make it all work on your first go around for your first drug. And we've seen some companies try to do that in this space, and I won't call out specific companies, but there's been failures. There's been one thing to be aware of is that there is no FDA approved drug yet that's been, let's say, primarily discovered and developed using AI. These companies have been around for 10 years now. AI is a loose term. We could have a whole podcast around how do you define what is AI versus machine learning. I'm not going to go down that rabbit hole or pull you, Tabari, down that rabbit hole with me. That would kind of
we'll do that
boring.
later.
We'll do that another time.
Later.
But assuming that people generally understand what AI is, let's say it's been 5-10 years of deploying this for drug discovery, and there's no primarily AI-driven drug that's on the market. So all of us collectively need to get to that stage first. And I don't even care if it's my company. One big milestone for our space will be someone gets there. There are drugs in phase 2. There are drugs that look interesting. Not there yet. Of course, again, as I said, then you have to get into, well, when someone says it was primarily AI discovered and developed, what does that really mean? Did they put a little veneer on? It's like when they say made in and you fill in the blank of the country and they put one button on the shirt and then it's made in this other. So that's a whole nother topic as we talked about. But I think that's a milestone for us. So when that happens, even though it probably won't be our company leapfrogging a large number of other companies that are already in clinical development to get there, it's unlikely that would be us. But when that other company does it, I'll be cheering, because I think that will change the perception out there in the world as to what's possible with AI. Until then, the naysayers that say, hey, this is all a bunch of hype, there's at least a little bit of truth to that in terms of where we are. Not that this won't all happen, but in terms of how far we are along today. So that applies to my company. That applies more broadly to the space, the AI drug discovery space.
Yeah. I think you're right. A rising tide is going to lift all boats, right? And so as long as we get someone to the finish line, I think that's going to open the floodgates for other companies to be thinking in this way and to realize that their success and to kind of point their compass in that direction, maybe not full on, but at least instead of going straight north, they're now going northeast, right?
Right.
So would love to talk about, I come from the medical affairs side, right? So I come from that kind of transition between R&D to commercialization, and really from and additionally from the kind of post-market evidence space, which I think is going to be crucial for any of these successful AI companies like yours moving forward, is having real clinical data and even potentially real world data or real world evidence to help validate models or to help build models. Do you see a future where the medical affairs organization is helping transition data into companies versus out of companies as they're doing right now?
Well, yeah, it's interesting. I'll give you a quick anecdote pertinent to that. So at ASCO, I was invited by Sean Cozen, who's the CEO, on the CEO Roundtable for Cancer, to a dinner where he had executives from across pharma in there, and there was more than one Med Affairs global executive. So top executive in Med Affairs for top 10, top 20 pharma companies, few of those. One of these guys was outspoken in his opinion that Med Affairs should play a central and leading role in AI in pharma. And what he meant was there's multiple aspects to it. You were just asking about one slice of that, which is data coming in versus out. I would say yes. But more broadly, the fears that people have about the use of AI in drug discovery, the application to specific disease types, the solving of problems for specific cohorts of patients with unmet need. I could go on down the list, and a lot of it would mirror what we've talked about over the preceding minutes here. But his point was Med Affairs has something to say about all of that and should be the one of, if not the central hub nodes again for all of this discussion, because you face inward towards your company and the science that's going on in the company, the clinical development that's going on in the company, but you also face outward towards the patients and the physicians that are writing prescriptions for these medications. There's only really one function that does all of that. That's Med Affairs. So I'm really borrowing or stealing from what this guy said, but he took this strong opinion and then eloquently explained it. I'm doing maybe a B+ job of replicating what he shared, but it made a lot of sense to me as somebody that's not an expert in Med Affairs myself, that when he described the function of Med Affairs, the different constituencies, and said, look, since AI is going to reinvent everything in pharma, Med Affairs has to play a central role. So that's my broader answer about Med Affairs and this whole AI drug discovery set of developments that we've been talking about.
Yeah, thank you. Maybe to talk more about future and AI, do you think that AI-driven R&D and commercialization across pharma becomes the standard? Is what you do going to become commoditized in the next 30 years, or are you still going to be, you and Archetype still going to be kind of that different R&D drug discovery company?
Well, Tabari, 30 years is a really long time. So, one thing I'll point to is if you're I don't know if you're familiar with Ray Kurzweil and the singularity, this concept of at some point we'll have artificial general intelligence and so on. He marked that at 2048, I believe, which is 23 years from now. So 23 years, 30 years from now, I don't know what our world's going to look like that far out, but even 5 to 10 years out, AI will touch almost all facets of pharma, from the earliest research all the way to what's happening as that, hopefully it's still a rep, but maybe it's a bot or a robot or something else is detailing a clinician. That whole process is going to be massively impacted by AI. I think the question, so first of all, there's no question that that's true. The question is timescale for that to ubiquitously go across. And the second question is, which pieces will take it on more substantially earlier? So is the next thing going to be that a robot is detailing a doctor, or is the next thing going to be that we're doing more automated preparation of FDA filings? Probably the latter. So I think that's the question, is this will happen. It will be ubiquitous, as it will be throughout all of our lives across all industries. Some of the things that's already happening is in the space that we've been talking about. So we're not the only company. There's dozens of companies doing different things. So will Archetype or an Archetype-like company stand out doing exactly what we're doing today 20 years from now? Probably not. There will be other capabilities that'll come online. The pace at which algorithmic stuff is coming online is unfathomable. We are, I think, towards that elbow of the curve that Kurzweil talks about in the singularity. We're there with AI in general. Now, as you know, when you enter a really steep curve on a road, at the beginning it's straight. So you don't know it's a curve until that curve is right on top of you. I think we're almost at that moment. Is that 1 year or 5 years? I don't know, but it's more 1 to 5 than it is 20 to 30. So we're much closer to that. And then I think that none of these companies that exist right now, if they exist in the form doing the stuff they're doing now, are going to be standouts. They're going to have to evolve massively. And it's really going to be about the interface of the AI and the problem you're solving, not just the AI. The AI that exists right now will be very old hat very quickly.
100% agree. Just in 6 months, I've gone from an AI novice to probably an AI intermediate, meaning I now have agents doing things for me, right? Which is fantastic and phenomenal. But I can only imagine what this looks like when agents are talking to agents, or when we get to a place where you, Tom, say, I need a new target therapy for some rare cancer that we've just found. Screen a billion molecules, and you just say that out loud, and then all of a sudden you get to that 10 we talked about, versus having to even have a team to do that. I think this is a great time to be alive as a human. It's also scary, but I think that we as clinicians and scientists and biotech executives have an opportunity to really change the course of human health using artificial intelligence. And yes, I am afraid of the singularity. So I'm a little bit concerned about that, but hopefully we'll be fine. Well, Tom, maybe I'll give you 2 last questions, and then we'll close up here. So would love to hear you opine on the roadmap for next-gen generative chemogenomics.
Yeah, I think the first thing for us we talked about a little bit is in our world specifically, the validation of our approach has partially been addressed by the things we talked about, the AACR presentation and so on. But ultimately, the real validation is that a drug that's been produced by what we described earlier in the discussion actually works in patients. So part of the roadmap, it may not matter so much whether we are running the clinical trial or whether we out-license a molecule that a pharma brings into a clinical trial. I don't know that that's so critical for our roadmap, but it's fairly critical that there is validation that this approach produces molecules that actually help patients. Otherwise, this is all an AI hand-waving exercise, despite all the validation data that we already have. So that is high up on the roadmap, and we have to put ourselves in a position to ourselves or through a partner make that happen. Beyond that, the next things that are on the roadmap are the expansion from a couple diseases to many diseases and making that scalable. How do we ingest the data and do all the pre-work to set up the AI to be able to learn these models? And then on the back end of that, how automated is the deployment of the insight? We've been talking about, hey, there could be a world where people come to the Archetype vending machine as a pharma company, and they say, I want a drug for stage 2 patients that look like this. I'm going to upload my TPP into the vending machine, and I want molecules that have this kind of IC50, and I want these other properties. I turn the knobs, press the button, and out drops a set of molecules that on the back end, someone at eMolecules or Enamine or somewhere else then ships directly to the pharma on a 386 well plate, and they can do their own testing, or we can do the testing and get it right through. So the automation of the action on the insight. What I talked about earlier in the talk is how do we get to the 10? Well, you then have to test the 10. You can start to integrate that into this process. So that's on the roadmap. I mentioned the expanding to many diseases also being on the roadmap. And then there's also modalities. We're in small molecule right now. That's just the way that the platform can learn. Getting to biologics, there's been a lot of pull from others saying, hey, would this work for a large molecule? Not today, but we know how we would do it. That's on the roadmap. That's already more than one little company can handle, but I'm just giving you a sense of the things that could be done. We're not going to do all of those things. We want to be focused. We're going to take those things one step at a time. But those are all really exciting possibilities that build on this framework that we've already established.
Thank you. Now, last question. I'm sure after hearing this, people are going to want to reach out to you specifically or to Archetype to understand how they can work with you. Is there somewhere I can send them or we can send them?
Absolutely. So I'm inviting a lot of email with this, but my email is out there at this point. So I can channel people to the right folks on our technical team for questions, or if they're talking business, it would be me or other people. Tom at Archetype, ARCHETYPETX.com. That's also our website. That URL is also our website. So that's how people can find out a little bit more about the company, or they can email me using that email address, and I will make sure their question gets to the right people in the company. I work long hours. I'm happy to play that role and with our admins and others to get the questions to the right people. And we're excited, as I mentioned earlier, to partner with people. So yes, of course, we want to work with pharma companies and investors and so on. But there's also lots of other AI companies and other companies, the data companies and others that have pieces of the puzzle. So I appreciate the opportunity to share that contact info and would invite anybody that's interested in learning more to reach out.
Awesome. We will put that in the show notes as well, both your email and the website for Archetype Therapeutics, so you can click there. Well, Tom, this has been a fascinating discussion. Thank you so much for joining me here on Scientifica Sessions. I am excited about the future that you're generating. So if there's anything that we can do here to help, would love to help you with that. But thank you so much for joining us today.
Thank you, Tabari. It's been a great discussion.
Thank you for listening to Scientifica Sessions, unscripted conversations with those shaping the future of biotech. If you found value in today's discussion, be sure to follow and subscribe on your favorite podcast platform so you never miss an episode. We release regular conversations with leaders across biotech, pharma, and medtech, all aimed with helping you stay ahead of industry trends. And if this resonated with you, please consider sharing it with a colleague or your network, because the more voices we can bring to the table, the stronger our community becomes. So until next time, keep questioning, keep learning, and keep shaping the future of life sciences.